that we've given time for people to arrive in the hall and for people to transition from one session to the other. I'm going to call on Sean to give the first talk. Sean.
All right, thank you very much. I would like to thank the organizers and Dr. Olapati for inviting me to give this talk today. In the past, I had some competing interests, which I think are all resolved at this point, and so I can just move forward with a scientific presentation. I'm going to cover two topics. I'm going to spend about a quarter of my time on a general overview of cancer susceptibility genes, and then I'm going to spend about 3/4 of the time on sequence variant classification. So, on to the cancer susceptibility genes. It's now more than 150 years since Paul Broca published a description of his wife's multi-generation cancer pedigree. But today, whether a physician is seeing a proband from a multi-generation high-risk pedigree like this, or perhaps an individual breast cancer case without any relatives who are clearly also affected, but with a couple of children. It's the case today that either way those patients, and even more so their at-risk relatives, may benefit from cancer predisposition testing. When I'm starting to learn about a new cancer susceptibility gene, there are really three sorts of things that I like to know about early on. The first has to do with the tumor spectrum that the gene is responsible for. And this slide is really focused on breast cancer and colorectal cancer genes. The second thing I'm interested in is, what is the magnitude of risk conferred by pathogenic genes in the gene, or pathogenic sequence variants in the gene of interest? And the third thing is, what is the summed frequency in the general population of the pathogenic variants in that gene? And so these days we sort of talk about three groups of genes in that sense. There are the high-risk susceptibility genes, where pathogenic alleles are conferring between 5 and 10-fold or even higher risk of the cancer of interest. But the summed frequency in the general population of the pathogenic alleles in those genes is not more than about 0.2%, 2 persons per 1,000, not in the cancer case population, but in the overall general population. The second group of genes are the moderate risk genes. Pathogenic variants here confer 2 to 5-fold increased risk of disease, and the summed frequency of the pathogenic alleles in those genes can go a little bit higher, maybe to about 0.5%. Dr. Kurian will be talking about moderate risk genes in her talk. Then the third group of genes that we talk about a lot now are the modest risk SNPs, which are extremely numerous. The allele frequencies for moderate risk SNPs can go right up to 50%. The risk conferred by the individual modest risk SNP alleles is only on the order of 1.1 to 1.25-fold. But there are so many of them in the genome that individuals can inherit a disproportionate fraction and end up at sort of increased risk for that reason. And Dr. Garcia-Closas is going to tell us about how one calculates and uses a polygenic risk score based on those modest risk SNPs. Another thing that's important to know about is what fraction of the familial relative risk of a cancer is explained by the known susceptibility genes. And in the case of breast cancer, we know that the high-risk genes, BRCA1 and BRCA2, plus the less common high-risk genes like P53, PALB2, PTEN, and STK11, together account for about 25%. The moderate risk genes account for about another 8%, and the SNPs, as a polygenic risk score, account for about 18%, which means that altogether, the known genetic risk factors for breast cancer account for, or the known genes account for about 50% of the familial relative risk of the disease. Okay. Now, setting myself up for the main topic of this talk, what is a variant of uncertain significance? A VUS is a sequence variant that, when observed during clinical mutation screening of a bona fide susceptibility gene, may be pathogenic, but is not necessarily so. Since common variants, those with a frequency of above 1%, and maybe even those with a frequency of above 0.1%, are generally classified early during the clinical development of a new susceptibility gene, this means that the VUSs are individually rare. And in general, VUS The largest proportion of VUS are missense substitutions. The next larger group are splice junction variants. Then comes in-frame insertions and deletions. And then there's other sort of complicated stuff like deep intronic variants that can interfere with splicing and so on. But even though the VUS are individually rare, they are really numerous. And this slide explains why. So we start out by knowing that recent whole genome sequencing studies in pedigrees have shown that the nucleotide substitution rate per generation in people is about 1.3 times 10 to the minus 8 substitutions per nucleotide across our genome per generation. That's a small number. And in fact, because there are rate across site differences, that is, there are a few nucleotide positions with really high rates, and most nucleotide positions have lower rates. The median rate is probably closer to 1.1 times 10 to the minus 9 substitutions per site. That's a really low number. But, the size of the human population is 7 times 10 to the 9th individuals. And that means that in one generation, the human population produces 7 substitutions per site across the whole genome in our gene pool. We, humanity, are a saturation mutagenesis experiment now in every generation. And therefore, the majority of all possible substitutions already exist in the gene pool, and almost everyone in the population carries one or more very rare missense substitutions. That is to say, almost all of us have variants of uncertain significance. So now, moving along to sequence variant classification. In 2008, I convened a meeting at IARC, the International Agency for Research on Cancer, to develop guidelines for classification of VUS and cancer susceptibility genes. The guidelines that we developed were both qualitative and quantitative, and they featured probabilistic classification thresholds defined as posterior probabilities in favor of pathogenicity for the clinical categories such as likely pathogenic and pathogenic, or likely benign and benign. But the system that we developed was not efficient enough to keep up with the rate of sequence variant discovery that happened once, for instance, panel testing started to become common. In 2015, the American College of Medical Genetics published their guidelines. The ACMG guidelines are qualitative, and in my opinion, a lot of other people's opinions, the qualitative guidelines are somewhat Byzantine. They acknowledge probabilistic classification thresholds, but nowhere in the guidelines are the probabilistic thresholds actually used. On the other hand, the ACMG guidelines are operationally more efficient than the IARC guidelines ever were, and trumping everything, the ACMG guidelines are FDA approved. To give you sort of a feeling for how those ACMG guidelines work, the first thing is that they have several levels of strength of evidence. They're qualitative labels like very strong, strong, moderate, and supporting, and a lot of different kinds of data that fall into each one of those levels. So for instance, there's a very strong piece of evidence that is a null variant, and then strong evidence can be things like amino acid change at a position where there's already the same amino acid change is one that's already known to be pathogenic, or a well-calibrated functional assay. Okay. Following those evidence criteria, there are qualitative combining rules that tell you which combinations of evidence reach the classification thresholds, like pathogenic or likely pathogenic. So for instance, there can be a rule that says that one very strong piece of evidence plus one strong piece of evidence equals pathogenic, or one strong piece of evidence plus 3 moderate pieces of evidence equals pathogenic, and so on. It's this combination of the evidence criteria and the combining rules that together is just more or less Byzantine. So we wanted to move past this. And in the next couple of slides, I'm going to show you some math. The point of view of showing you the math is not that I want you to understand anything about the math. It's actually to get the slides that are 3 slides from now and 4 slides from now that turn the thing into a simple point system. But I can't get there without giving a justification. So in 2018, we were able to fit the entire ACMG qualitative system into a single Bayesian equation given here. But the problem with that equation, even using that equation, is that across the diversity of math skills among the variant curators who actually do the variant classification, using the equation is just too complicated. It's not a viable option. So then, with a little bit more math, we were able to see within the classification equation an incipient point system that's given in the bold-faced numbers at the bottom of, in the last equation down here, the 1, 2, 4, and 8, and the -1 and -4. And from those, we're actually going to get a natural point system that you'll see on the next slide. The reason that a point system can pop, the reason that the Bayesian system popped out and then the point system with these evenly scaled numbers is actually that within the ACMG combining rules themselves, there was repeated use of a rubric that is basically 2 evidence criterion at a given strength, like moderate, is equivalent to 1 evidence criterion at the next level, like strong. Richards et al., the team that wrote those rules, did not know that they put that rubric into their system, and yet the majority of the rules are related to each other by exactly that rubric, which isn't even evident until after you've done the curve fitting, and then look back and say, well, why did that work? Well, it worked because there was this rubric that was systematically part of the rules. In any case, after the little bit of math that's been done so far, and then making a couple of simple assumptions, we get a point system. And what the point system is that very strong, moderate, and supporting evidence are worth 8, 4, 2, and 1 points respectively. And then on the flip side, the benign evidence is worth -1 times whatever the pathogenic evidence was. These points are log odds, and therefore they are additive. Beyond that, what I want to be very clear about is that the points are mathematically derived from the ACMG rules. They're not arbitrary, they're not being pulled out of thin air, they're actually related directly back to the rules that were published in 2015. We can do a little bit more algebra and then extract the classification thresholds also from the same equations. And so the thresholds are that pathogenic is a score of more than 10 or more points. Likely pathogenic is 6 to 9 points. Uncertain is 0 to 5. Likely benign is -1 to -6 points, and benign is less than or equal to -7 points. Though in the likely benign case, it's probably better to use -2 points as the threshold rather than -1. And of course, since I said that the points are log odds and add up, if you have various evidence criteria, with 1 point, 2 point, 4 points, or whatever, that add up to 6 or 7 or 8, then you're in the likely pathogenic range. If you get to 10, then you're in the pathogenic range, right? So it's as easy as, A, B, C, 1, 2, 3, do, re, mi. It's just addition and subtraction. Everybody can do it. So now, it's been expected for a long time that functional assays were going to be the key to being able to classify most of the unclassified variants. In fact, that was written in the discussion of one of the BRCA1 confirmation papers back in late 1994 or early 1995. And we are now getting to the point where this is going to happen. So I'm going to take a few minutes describing a particular functional assay, and then I'm going to show you how the classifier works. In the case of BRCA1 and BARD1 form an obligate heterodimer. Heterodimerization happens as a protein-protein interaction between the ring domain of BRCA1 and the ring domain of BARD1, which are at the end termini of both proteins. This being the case, we set up to do a comprehensive assay of all possible missense substitutions in the ring domains. To do the assay, we set up a mammalian 2 hybrid system, and I set it up as a 2-color, with a 2-color reporter to get better signal-to-noise ratio than could come from a single reporter, which is sort of the normal way of doing the mammalian 2 hybrid assays. All possible missense substitutions were array synthesized in vitro, but we synthesized each possible missense substitution in 4 different flavors using sort of a barcode of silent substitutions located nearby in the BRCA1 coding sequence that would let us distinguish between independent syntheses of the same missense substitution, and therefore know that in the actual experiment that we had observed each missense substitution 3 or more times as independent replicates before we could try to evaluate or calibrate the assay. Then our assay only took a little bit of laboratory automation and a lot of elbow grease to get through all 700 possible missense substitutions in the ring domain. Now, basically what happens in the assay is that all the cells for a single experiment get combined and flow sorted. And then, if a missense substitution is resulting in relatively high activity of both reporters, the flow sorting puts it far out on the red-green axis. Whereas if a missense substitution is dysfunctional, then the flow sorting leaves it close to the origin on the red-green axis. We pool together all of the cells that are flow sorted into these individual bins, sequence RNA from those cells, and thereby associate each individual missense substitution with its level of functionality. Here is an individual flow sort for a benign sequence variant where you can see lots of cells far out on the XY axis. And I can tell you that the benign missense substitutions flow sort identically to wild type. Here is a well-known pathogenic missense substitution, and you can see that its cells don't get nearly as far out on the red-green axis. Now, I am just going to breeze through this because it's not really important here, but it took us 7 individual experiments to cover the whole ring domain with each individual substitution observed 3 or more times. But at the end of that, we were able to, at the end of the data collection, right, we knew the level of activity of each individual missense substitution, which is every possible missense substitution. Within the list of every possible missense substitution, there were 29 that had significant patient observational evidence in favor or against pathogenicity. This is evidence from personal and family history or evidence from segregation. Nothing coming from functional assays, nothing coming from computational data, so that there are no circularities in the connection to the overall sequence variant classification model. And in our analytic space, the sequence variants with strong personal, almost all but one of the sequence variants with strong personal and family history evidence of pathogenicity go in the upper left, and the ones with little or no evidence go in the lower right, except one variant that had sort of moderate evidence in favor of pathogenicity that grouped with the benign variants. Doing a whole bunch of math that's actually beyond me on this data, we're able to derive the contours that give the ACMG levels of classification, so that our dotted red line is supporting heavy dotted red line moderate and solid red line strong evidence in favor of pathogenicity according to the calibration of the ACMG classification scale, and then the inverse for the light dotted, heavy dotted, and solid blue lines towards benign effect. Okay. Also, within the list of all possible substitutions, there were additional 66 missense substitutions that had been observed in either a clinical series or a population database that's nomad that could be used for validation, and I'll tell you about this in a moment. But right here, the ones that are in red were observed in clinical only, the ones in blue were population only, and in gray were clinical and population. So you can see already that the red ones end up on the upper left and the blue ones in the lower right. Plus all remaining substitutions, and you can see that the vast majority of possible substitutions are looking benign. There's a clear subset that are looking pathogenic, and only a small number in the middle. Okay, we've got to validate this. We had data from roughly 200,000 clinical subjects and 200,000 population subjects from nomad, and because from those, there were 66 sequence variants that were observed in either case or control, from which we can calculate odds ratios. To avoid any circularity, the 29 calibration variants are excluded from this analysis. And because these are very clinical cases which result in an exaggerated, what we call pseudo odds ratio, to give you a scale for what we're working with, I can tell you that the pseudo odds ratio for truncating variants in BRCA1 by the same analysis comes out as 34. And for BRCA2, it comes out as 18. And so the pool of missense substitutions ended up in the upper left of that flow sort have a pseudo odds ratio of 18.6 associated with them. So that pool, on average, is like truncating variants in BRCA2, but maybe not quite as severe as truncating variants in BRCA1. On the other hand, the missense substitutions that ended up in the lower right had an odds ratio of 1, right? That's indicative of sequence variants that are a pool of sequence variants that is well and truly benign, which is what we need. Okay, finally, we get to the fun part. We're going to apply the classifier. So what I need is I step into what's an ACMG compliant classifier using data that are available for every single missense substitution, is that you need to know that this vertical blue line here is the likely benign threshold at -2 points, and this vertical gold line is the likely pathogenic threshold at +6 points. I'm going to combine together 5 kinds of data to walk through this. The first piece of data, the first datum, is whether an individual missense substitution is falling at a canonical residue in the ring domain, at a residue that's critical for the interaction between BRCA1 and BRCA2, or at a different residue. Those guys are getting 0, 1, or 2 points in the point system. Then we are going to add in the next ACMG data code, which is whether or not a missense substitution is falling at a residue where another missense substitution is already known to be pathogenic. That's worth 2 points, and it basically splits these categories. So for instance, the categories that were previously at 2 points is now split. So some of the variants have 2 points and some of the variants have 4 points, okay? But we're starting to spread the variants out a little bit. Then we add in computational tool, a computational prediction of whether the sequence variants are pathogenic or not. And so now the sequence variants, the missense substitutions with the prediction of pathogenicity are turning orange. Those with a prediction of benign effect are turned blue, and those with an indeterminate prediction stay gray. No sequence variants have crossed the likely pathogenic or likely benign thresholds yet, but they're actually starting to get spread out pretty nicely, so that when we add in the functional assay, lo and behold, a large fraction of the missense substitutions jump across the classification thresholds, with a group into the likely pathogenic range, a big group in the likely benign range, and only about 15% of the missense substitutions left in the unclassified interval. As a last step, we can add in the little tiny bit of observational evidence that would be available for any clinical observation, which is whether the sequence variant is observed in nomad-only population, observed in the clinic but with no population observations, or observed sort of in both places. And those data spread the variants out a little bit more, with a few moving into the pathogenic range and a few moving into the benign range, okay? Well, it's just to say, we've really got this now. In the case of BRCA1, using data that are universally available for any missense substitution in the gene, we can now immediately classify about 85% of missense substitutions. What's required to classify even a larger fraction of them is data that the clinicians hold. It's actually the personal and family histories, or else it is tumor immunohistopathology data. So it's really important to gather those data and pass those data along to the testing companies where they can be combined in to the variant evaluation. The last thing to do is to show you that, in fact, we've got evidence in favor of that classification. So going back to my odds ratios now, we can look at the sequence variants that have a score greater than or equal to 6. And their odds ratio is now 33.6, which is identical to nonsense substitutions in BRCA1. And the, I'm going the wrong way. And the odds ratio for sequence variants with a score of less than or equal to -2, which is the likely benign group and benign group, remains 1. So that's clearly a group that's nothing but benign sequence variants. So in summary and in conclusion, my first little bit of summary here is sort of more general, even for the whole session, than having to do with my talk, okay? Most of the benefit of genetic testing for cancer predisposition genes really goes to the at-risk relatives. There's more and more patient treatment benefit accumulating as we understand more about these genes and more about the effects of, for instance, PARP inhibitors. And most of that benefit is going to the at-risk relatives. In order to realize that benefit, it's really critical to emphasize cascade testing of at-risk relatives once a pathogenic variant has been observed. And beyond cascade testing, it's even more important to enable prevention, early detection, and cancer interception in the at-risk relatives. If those last two steps don't happen, then the genetic testing is practically for naught. Secondly, the last 5 years has seen considerable progress in the problem of variants of uncertain significance. The point system that I showed you is derived directly from the ACMG variant classification system. It's not arbitrarily pulled out of thin air. It's really a mathematical derivative, and it relies on a very sound foundation of Bayes' rule. It's just addition and subtraction, so it's easy to do. It has the advantage that qualitative evidence criteria can be slotted in by expert opinion, and quantitative evidence can be added in by empirical calibration. And also, I would say that over the next couple of years, there's going to be a real effort to check the calibrations of the expert opinions that were provided by the ACMG back in 2015 to see where they're correct and where they might need a little bit of modification. Comprehensive functional assays are being calibrated and are almost ready for general use. For BRCA1, the job is basically done and it's just going to be refined a little bit. For BRCA2, I'm pretty sure that the comprehensive assays are going to be done next year. And probably it'll be the mismatch repair genes for colorectal cancer that follow after BRCA1 and BRCA2 are done. But one thing that you may have noticed from the sort of pyramid diagram that I showed you is that it's really concordance between the computational tool result and the functional assays that's yielding efficient classification. When the computational tool and the functional assay are concordant pointing benign, then the sequence variants end up as likely benign, and when they're concordant in the pathogenic direction, the sequence variants end up in the likely pathogenic bin. And that brings me to my new mantra, which is based entirely on the point system, which is just that, 2 + 4 = 6. So you better make sure that the 2 is correct, that the 4 is correct, and addition is the correct operator. Okay, what that really means is that 2 points is what gets assigned from well-calibrated functional tools in papers that will be appearing in the next 6 months. 4 points is what can be assigned from a well-calibrated functional assay, so you've got to make sure that those calibrations are correct. The operation of addition in the point system rests on the assertion that the various kinds of data that are being combined are conditionally independent of each other. And so it's important over the next couple of years to actually test the assertion of conditional independence, which is being tested in a paper that I have under review right now. And thus, 2 + 4 = 6. So let's make sure that the 2 is correct, the 4 is correct, and addition is the correct operator. Thank you so much for your time. I'm not sure that I'm supposed to take questions right now, or whether we wait until the end.
We have time for a few questions. That's really wonderful. And for those of you in the audience, we have microphones in the aisles. And then for those of you who are watching us virtually, please put your questions in Slido, and they will be sent along. And so I'm going to start by saying it's really wonderful to be able to educate us on how we make sense of the sequence, because you can't do genetics and do genetic testing and give results back to patients without actually understanding what goes on behind the scenes. So my question is, I have two from your initial discussion. When you started off, you started talking about breast and colorectal cancer genes. Why is it important for us to think about these genes in terms of ordering a test and trying to do high throughput panel sequencing?
Okay. That is a super great question that has become, the answer to which has become clear in the last, say, 4 or so years, which is that there are breast cancer genes, things that are called breast cancer genes, that also confer moderate or even greater risk of colorectal cancer. I mean, it's clear that, for instance, BRCA2 is conferring some increased risk of colorectal cancer. And the colorectal cancer genes, some of them are conferring increased risk of breast cancer. In particular, MSH6 confers increased risk of breast cancer, right? P53 is a susceptibility gene for both. And so there's considerable overlap now in the tumor spectra conferred by susceptibility genes. And it's even more complex and a greater indication for why the full panel tests need to be run when one looks at which genes are conferring increased risk of ovarian cancer or pancreatic cancer or any of a large number of cancers.
Okay, so and the second question I have for you is there's so many genes, and we're doing this panel and that panel, and the expansion in terms of the different cancers that we have to think about. So when you think about probability, right, why is this variant important or not important? How do you explain statistics and probability to the patient who's just asking for a yes or no answer?
So,
because you've given us higher order mathematics now, but we're going back to the clinic on Monday to talk to the patient about you need to act on this mutation.
Right. So I think that the most useful thing that the most useful formulation of this that the genetic counselors and clinical geneticists can do is when they convert what I was calling odds ratios into 5-year and 10-year risks. Those 5-year and 10-year risks are really telling the patient what they should expect in the not too distant future and help inform the patient about what sort of risk reduction and disease prevention strategies are most appropriate for them. And sometimes you see these diagrams that, show you a field of people, and then you can have some proportion of them change to a different color to give you sort of a more pictorial representation of what those 5-year and 10-year risks actually mean.
Yeah, so
but that's
great.
my impression of what works well for patients.
Okay, very good. We'll take one question from the virtual audience, and then we'll come to the speaker. So the question is the ring finger heterodimerization an example of a functional assay, of the functional assay when it comes to BRCA1? Is it an example, or is it the only functional assay that we should care about?
So that's a fun question. 2 years ago, there was a high throughput viability assay in haploid sort of stem cells that was published by Jay Shenduri's group. And that was an assay of 95% of all possible nucleotide substitutions in the ring domain and the BRCT domain of BRCA1. And so that assay is almost comprehensive, and its results are quite useful. I personally am a little bit skeptical of the calibration of that assay, but the second point to make about it is that I'm going to be careful to do a two-sided explanation of this in the next few sentences. On the one hand, the viability assay is not in any sense mechanistic. We don't have an understanding of how viability in that particular cell line or any cell line is related to cancer susceptibility. And there could be things about that viability assay that are not predictive of what's really going on in patients. On the other hand, the assay that we have done, the mammalian 2 hybrid assay, it's a mechanistic assay. We know that it is testing a functionality that is critical for tumor suppression. There's a small possibility that there are other functionalities in the ring domain that are also important. There's also a possibility that because the cells that we use for the assay are not breast or ovarian, I should say breast or, there goes my vocabulary. Tubes.
Fallopian tube.
They're not breast epithelial or fallopian epithelial cells, that there could be some slight intracellular difference that's making the assay perform differently than it would in the absolutely correct kind of cells. Nonetheless, it's a mechanistic assay, and I think the two of them together provide a better picture than either one of them can alone.
Okay, very good. So yes.
Hi, Jessica Jones from Houston. So earlier today, we listened to some talks in the hall giving us a call to action to address disparities in breast cancer and breast cancer research. So recently, there's the paper release that talks about how oncotype DX, or the gene mutations on the breast cancer are, even though an African American woman will have the same score as a white woman, she will still do worse or have a higher probability in that recurrence risk. So knowing that, looking at these variants of unknown significance and looking at the 200,000 clinical subjects that were utilized, do we have like a background? What were the different ethnicities of women? And on that note, is the mechanistic function, does it differ among African American women?
Right. Okay, so that's a nice two-part question. The first part of it is one thing that came out when I was looking at these data was the stark difference in the proportion of patients of ethnicities other than Western European in the patients versus the population database from NOMAD. The patient population was dramatically more Western European, dramatically less African American or Latino Hispanic than the NOMAD population. And this is a consequence of access to testing these past 20 or so years, right? There's a disparity, there has been a disparity in access. I hope the disparity in access is being reduced. I'm not convinced that that's the case yet, and I would definitely do everything that I can, which is not a huge amount, but still everything that I can to help reduce that disparity. Now, part of the point of doing the comprehensive functional assays is to evaluate every possible sequence variant. When we evaluate every possible sequence variant, we take anything about race ethnicity out of the picture, right? Because we're all humans, we have the same basic sequence, and that's what there is to evaluate at this level. But still, there can be differences in outcome based on race ethnicity, which maybe are more due to socioeconomic status and access than to real, true underlying genetics, and which are going to be hard to erase. Fumi?
Okay, yes.
If you have extra
Yeah,
words
so
to add to that.
we'll bring that back to our panel discussion because I think that's an important question to bring to the entire group. So we'll take one more question from the audience here, and then we'll go to the next speaker.
Thank you. So this is Seema Khan from Chicago, Northwestern. And I'd like to thank you for a really elegant discussion of your model and the combination of computation and biology. It's a great place to start because BRCA1 and BRCA2 are obviously are among the common alterations that we encounter, but you talked about panel testing, and the problem of variance, I think, in the other genes that are on commonly used panels is in fact a more prevalent problem at the moment. So how do you envision applying these systems to that, expanded set of genes? But before you start your answer, I'd also want to thank you for mentioning the importance of short-term risk estimation and the value of using that to counsel patients, because I entirely agree with you.
Yeah.
Okay.
So I hope you heard that question.
Yeah.
Yes.
First of all, thank you for the last part. It's absolutely the case that there are unclassified variants in practically all of the genes, and that it's going to take a while to get to all of them. And so I think that the strategy is to start with the highest risk genes. Back at our meeting in 2008, Doug Easton made the point that the probability that a variant is actually going to be pathogenic is, our first estimate is the sequence variant probability of pathogenicity, which we can do now, times something related to the odds ratio of the gene. And that automatically means that the unclassified variants in the high-risk genes are more dangerous than the unclassified variants in the moderate-risk genes. And therefore, there's a strong justification for starting with the high-risk genes. I can say that P53 is actually all largely done. That's the highest risk gene. BRCA1 is basically done now. That's the second highest risk gene. BRCA2 is going to be the next one up. Then after that is going to come CHEK2, ATM, and PALB2. It's It's going to turn out that for PALB2, almost no unclassified variants are actually pathogenic. It's going to turn out that for CHEK2, a really large fraction of the unclassified variants are pathogenic. And it'll be maybe 2 to 3 years before the functional assays get to the bottom of those, but it's going to happen.
Yeah.
For the mismatch repair genes, there's already good progress, and that's going to continue for the next couple of years.
Yeah. So actually, that's a really nice segue to our next speaker. Thank you